A cross-section of the cycus stem shows some roughly circular outline because of the persistent leaf basis. Here you see the tiers of stem of Jamia fluidiana. Here you see the massive cortex and massive pith. This massive cortex and pith is interacted by persistent mucilage canals. This here you see that endodermis and pericycles are not very clearly distinct in the stem of the Germia. Here you see the primary vasculature is conjoint, collateral, open and dark. Here you see the leaf traces. The leaf traces. These leaf traces are originated in the bundle, the vascular bundle near the vascular leaf traces or near the vascular primary vascular bundle and in the base of the petiole or also in the distal portion of the vascular bundle. From here, these leaf traces enter the petiole. First, it traverses horizontally in the vascular bundle, then it moves directly opposite before entering into the petiole. Here you see this type of leaf stress arrangement is known as the girdling bundles. It is the characteristic feature of the family Cycadales. Here you see the secondary xylem is present in very little amount. This is good for this because of this very little amount of secondary xylem. The persistent leaf bases sclerenchymatous tissues give strength to this monoxylic wood, which is soft-wooded trees. This sclerenchymatous tissues of the leaves give strength to these stems of these members of Cycadales. Here you see the secondary growth is single, second is present in a single ring. This is known as the single ring of secondary growth, which is due to the single cambial ring present in the vascular bundle. So this type of arrangement is known as the monoxylic because there is only one ring of secondary wood is present, whereas in the other members such as cycas, macrogamia and encephalotus, the secondary wood is polyxylic. There are 2 to 3 rings of secondary wood are present in the stem due to the succession of the cambium in the stems of the cycadales. In the secondary wood, some rays are also present in the secondary xylem. Some rays are 2 to 2 cells wide, while some are very high. Some broader rays are also present through which these leaf traces move in the vascular bundle. In the family Jamia, in the genus Jamia and in the Strangeria, in the secondary xylem, scalariform pits are present, whereas in some other genera, only spirally circular bordered pits are present. Here you see the anatomy of the roots. In the family Cycadales, there are two types of roots are present. That is normal root and the coralloid roots. Normal roots are Normal roots of cycads are very thick at their bases and they are become thin towards their apex. In the young roots, the steel is diar, where in the mature stem it is diar to polyar conditions are found in the Cycadaceae. Here you see that there is outer epiblema, then it is below this epiblema you see the cortex region. Below cortex region, here you see endodermis and pericycle, and this is the steel inside it. This is the normal root of Cycadaceae. Whereas in the coralloid root, you will see that in the cortical region, here you see the triar conditions of steel, and this is the xylem. Three pairs of xylems are alternating with phloems. Here in the cortex, you see here some blue-green alga is present, such as anabina. These cells in which anabinas are present are slightly larger than the other cells of the cortex. So this is the main difference between the normal root and the coralloid root. Coralloid roots are negatively geotropic and it is just just looks just above the ground. While these normal young roots, these are tap roots and they grow towards the gravity. Here you see the and the coralloid root. The coralloid roots looks like this. Here you see the cross section of the coralloid root. This is root surface. This is the colony of the cyanobacteria. Here, this is the coralloid root. This is also the cross section of coralloid root. Here in the middle, you see the triaxylum, inner cortex. This is the cyanobacterial jaw. This is outer cortex and periderm. This is the anatomy of the. This is the anatomy of leaflet of Cycadaceae. This is the leaflet of cycas. Here you see the upper. This is the upper epidermis and this is the lower epidermis. This upper epidermis and lower epidermis, here you see that the stomata are present only on the lower surface of the lower epidermis, and this type of stomata, here you see the sunken type of stomata. Here you see below this upper epidermis, you see the region that is hypodermis. This hypodermal region where the cells are very compactly arranged. Below this region, you see here the mesophil tissue. The mesophil tissue is divided into two cell types, that is palisade and spongy parenchyma. In this columnar, in this palisade tissue, see the columnar cells. In these columnar cells, chloroplasts are present. These round shaped cells. In between these hypodermal cells, some round shaped cells are visible. These are spongy parenchyma. Spongy parasome chloroplast cells are also present in the spongy parenchyma. In the vascular bundle of the midrib and some leaf presses, you see this. Then now you see the two types of xylem. So that condition is known as diploxylem. Here you see the centripetal and centrifugal type of xylem. In the centripetal type of xylem, it is triangular in shape and there is one protoxylem. Whereas in the centrifugal, you will see the xylem is arranged in the form of the semicircular. It is different. It is different from it from parenchymatous zone, and it looks like the secondary growth. It is given an idea of the secondary in origin, but it is associated with tracheids, with phloem. This is the anatomy of petiole. Here leaf traces, which is arranged in the form of omega. Thank you.